this chromatin, the 3D chromatin structure is one of the layers. The epigenetic layers. Epigenetics is the science that studies how the genes change expression without mutations, without genetic changes, right? And there are multiple layers. One of them is the DNA methylation in one dimension. Let's say in this DNA string, the DNA can be methylated. Another one is how the DNA is packed with histones, which would be like 2 dimensions, I would say. And the 3 dimensions is we just talk about the 3-dimensional structure of the genome within the cell nucleus. This intermediate 2-dimensional level is very important because it allows us to segment the genome into particular functions. So we have done this for CLL and also for multiple myeloma. In CLL, we published a paper last year in which we could determine all these chromatin functions in CLL, and we found a lot of chromatin activation, right? In multiple myeloma, the level of chromatin activation is even higher. We see a massive activation of multiple regions that are totally inactive in normal plasma cells and in normal B-cell differentiation. And suddenly myeloma, they open and they start deregulating the expression of myeloma-related genes. And in fact, the target genes of this chromatin activation belong to pathways, several pathways that have been described to be important in myeloma. So in a way, this chromatin activation seems to be a feature that unifies or that underlines all the different mechanisms that we have observed to be deregulated in myeloma. And what is important of these signatures of chromatin activation that are disease-specific is that they seem to be mediated by the action of a small set of transcription factors. So these transcription factors that become deregulated in the disease bind to several regions of the genome, activate regions, and lead to oncogene deregulation. If we think about therapeutic opportunities, we can either go to the genes that become deregulated or the pathways, or we can think, well, if the cause of that chromatin activation is the action of a particular protein that binds the DNA and opens it, can we block the action of the transcription factor, and can that reverse this overall chromatin activation pattern in myeloma and in CLL? We believe that could be the case, at least from the theoretical point of view. That would be a very good strategy to move forward.